Right now, in hospitals across more than 30 countries, doctors are deliberately placing live fly larvae onto human wounds. Not by accident, not as a last resort of desperation, as a prescribed, regulated, clinically proven medical treatment approved by the United States Food and Drug Administration in 2004 as a living medical device. The larvae clean wounds that have resisted every antibiotic modern medicine can produce. They remove dead tissue with a precision no surgeon can match. They kill bacteria, including several of the most dangerous antibiotic-resistant strains on Earth. And they do it faster and more cheaply than any alternative treatment currently available. The animal providing this service is the one most people try to kill the instant they see it. This is not the story you were told about the fly. The story you were told is about filth, about disease, about an insect that lands on food, crawls on garbage, spreads what it touches. Something to swat, something to repel, something to exterminate. That story is not entirely wrong. Some species of fly do carry disease. The harm is real. But the story is so incomplete that it amounts, in practice, to misinformation. The fly is one of the most ecologically indispensable insects on Earth. Her species pollinate crops that bees cannot reach. Her larvae decompose organic matter at a scale that no other group of organisms can replicate. Her genetic simplicity has taught humanity more about human biology than almost any other model organism. 6 Nobel Prizes in medicine have been awarded for work done on flies. This episode is about an insect we have treated as disposable, while quietly owing her some of our most important scientific and medical breakthroughs of the last century. The fly is not a nuisance. She is, in more ways than most people would believe, one of the quiet, load-bearing pillars of the living world. And we kill her reflexively millions of times a day without ever pausing to consider what she actually does. Chapter 1: The biology of the most underestimated insect on Earth. There are over 150,000 known species of fly on Earth. Scientists estimate the actual number may be closer to 1 million, most of them undescribed, unstudied, and in many cases unrecorded in any scientific database. Flies occupy every terrestrial habitat on the planet: Arctic tundra, tropical rainforest, high mountain deserts, the surfaces of glaciers. They feed on nectar, on fruit, on blood, on decaying matter, on other insects, on fungi, on nothing at all. This staggering diversity is the first thing to understand about flies. When we say the word fly, we are not describing a single kind of insect. We are describing a vast family of organisms with wildly different lifestyles, dietary habits, ecological roles, and evolutionary histories. The housefly you swat in your kitchen and the hoverfly pollinating flowers outside your window share approximately as much in common as you share with a mouse. Let us start with the body itself. A housefly weighs approximately 12 milligrams. She has 2 wings, unlike most flying insects, which have 4. Her second pair of wings has evolved over approximately 200 million years into a pair of small club-shaped structures called halteres. The halteres vibrate at high frequency during flight and function as miniature gyroscopes. They give the fly real-time orientation data about pitch, roll, and yaw. They are the reason a fly can fly upside down, land on a ceiling, hover in place, and reverse direction mid-air with an agility that has never been matched by any flying machine humans have built. Aerospace engineers study the halter system. They are attempting, with partial success, to reproduce its principles in small drone design. The fly has had 200 million years. We have had 40. Now consider her eyes. A housefly has approximately 4,000 individual lenses in each compound eye. Each lens feeds its own signal to her brain. The result is a visual field that processes time at a rate of approximately 250 frames per second. The human eye processes time at approximately 60 frames per second. This means a fly perceives the world in something close to slow motion compared to a human. A hand moving toward her at what feels to us like normal speed appears to her as a slow, deliberate gesture. She has ample time to calculate an escape trajectory and execute it before the hand arrives. This is why flies are so difficult to swat. Not because they are lucky, because they are, in a very literal sense, perceiving reality faster than we are. And her sense of taste. A fly tastes with her feet. When she lands on a surface, chemoreceptors on her feet immediately identify what she is standing on. Sugar, salt, protein, chemical signatures associated with specific foods. Before she has extended her proboscis, before she has touched anything with her mouth, she already knows whether the surface is worth feeding on. Her feet are sensory organs, more sophisticated than most invertebrates possess anywhere on their bodies. And finally, her life cycle. A housefly can go from egg to sexually mature adult in as little as 7 days. Each female can lay up to 500 eggs in her lifetime. This reproductive speed is what made flies so valuable to science. A researcher can observe multiple generations in a matter of weeks. Mutations can be tracked. Inheritance patterns can be mapped. Genetic experiments that would take decades in mice can be completed in months in flies. The animal most people consider a pest is, in terms of biological sophistication, one of the most remarkable insects ever produced by evolution. Chapter 2. Maggot Medicine. The wound treatment that outperforms antibiotics. In the battlefields of the American Civil War, a surgeon named John Forney Zacharias noticed something his medical training had not prepared him for. Soldiers with open wounds that had been contaminated by fly larvae were healing faster than soldiers with clean wounds. He wrote in his journal that maggots, in a single day, would clean a wound better than any agents at his command. He was writing in 1865. Zacharias was not the first to notice this. Observations of the wound-healing properties of fly larvae go back thousands of years. Aboriginal Australian healers knew it. Mayan medical traditions included it. Military surgeons across multiple wars had documented the phenomenon. But the medical establishment, almost without exception, dismissed it as folk superstition. The arrival of antibiotics in the 1940s seemed to end the conversation. With penicillin and its successors, who needed maggots? Then, in the late 20th century, antibiotic-resistant bacteria began to emerge, and physicians started looking for alternatives. In the 1980s, a physician named Ronald Sherman at the University of California began to study what happens precisely when fly larvae are applied to a wound. His findings were remarkable. The larvae, specifically those of the common green bottlefly, secrete enzymes that dissolve dead and infected tissue while leaving healthy tissue untouched. This process is called debridement. In conventional medicine, it must be performed manually by a surgeon with scalpels at significant cost and with a measurable risk of removing healthy tissue by mistake. The larvae do it with enzymatic precision no human instrument can match. They also produce antimicrobial compounds that kill bacteria directly. Sherman's research demonstrated that maggot secretions are effective against multiple antibiotic-resistant strains. including MRSA, the hospital-acquired infection that kills tens of thousands of people every year and that modern medicine struggles to treat. The larvae kill what the antibiotics cannot. And there is a third mechanism. The larvae stimulate the wound itself. Their movement on the tissue surface, combined with the biochemical environment they create, accelerates the growth of healthy cells. Wounds that had been static for months, unable to heal despite intensive medical intervention, begin granulating, closing, regenerating. The larvae are not just removing damage, they are actively encouraging the body to rebuild. In 2004, the United States Food and Drug Administration approved medical-grade maggots for clinical use. They are produced in sterile laboratories from pathogen-free parent flies, shipped to hospitals in small sealed containers, applied to wounds for 2 to 3 days, and then removed. The treatment is cheaper than extended antibiotic therapy. It is faster than conventional wound care. It often succeeds where every other intervention has failed. Today, maggot therapy is used routinely in hospitals across the United States, the United Kingdom, Germany, Israel, Australia, and dozens of other countries. It is particularly valuable for diabetic foot ulcers, one of the leading causes of amputation worldwide, for pressure sores, for infected surgical wounds, for burn victims, for any wound that has stopped responding to conventional treatment. Studies have documented amputation rates reduced by over 50% in patients receiving maggot therapy compared to those receiving only conventional care. Hospitalization times shortened, infection rates lowered, costs reduced. The fly has saved legs. She has saved lives. She has done it at a fraction of the cost of the pharmaceutical alternatives she competes with. And this is happening right now, in hospitals that people you know may have been treated in. With an insect, most of us would instinctively kill without thinking. The disconnect between what the fly is doing for human medicine and how we conceptualize her as an organism is one of the starkest in our relationship with the natural world. We have given her no credit. We continue to sell billions of dollars of fly-killing products every year in markets where her larvae are simultaneously being deployed to save limbs. She is not waiting for us to notice. She has been doing this work quietly in the background of medical history for as long as there have been open wounds and fly larvae in the same proximity. We only recently began to admit it. Chapter 3, The Pollinators. We never counted. When most people think of pollinators, they think of bees. This is accurate, but incomplete. Bees are the most efficient and most studied pollinators on Earth. They are responsible for the largest share of commercial crop pollination in temperate regions. But they are not the only pollinators, and in many ecosystems, including some of the most economically important ones, they are not the primary pollinators at all. The fly is. Begin with one specific example that affects nearly everyone. Chocolate. Cacao trees produce the beans that become chocolate. Cacao flowers are small, white, and grow directly out of the trunk and branches of the tree in complex clusters. They are astonishingly difficult for any insect to pollinate. Bees cannot reach them. Butterflies cannot reach them. Most pollinators ignore them entirely. The exception is a tiny insect called the cacao midge, a species of fly no larger than the head of a pin. The cacao midge and a few related species are essentially the only pollinators capable of navigating the geometry of the cacao flower. Without these flies, there is no chocolate. Global chocolate production, an industry worth over $100 billion per year, rests on the activity of an insect that almost no consumer has ever heard of, and that most gardeners would attempt to eliminate on sight: cacao is not an isolated case. Mango flowers are pollinated primarily by flies. So are carrot flowers, onion flowers, avocado flowers. In many regions, a significant proportion of flowering plants across tropical ecosystems depend on fly species that co-evolved with them over millions of years. Hoverflies, a group that visually resembles bees and wasps but belongs to the fly family, are now recognized as one of the most important pollinator groups in temperate agricultural systems. They pollinate strawberries, apples, raspberries, stone fruit. They contribute to the pollination of oilseed rape, one of the most economically significant crops in the Northern Hemisphere. In cold climates, where bees are inactive for significant parts of the year, flies are often the dominant pollinators. In high mountain ecosystems and at Arctic latitudes, flies pollinate almost everything that flowers. Entomologists have been quietly documenting this for decades. The public conversation about pollinator decline is focused almost entirely on bees, partly because bees are charismatic and culturally familiar, partly because the economic data on managed honeybee populations is more complete. But fly populations are declining too, in many cases faster than bee populations, for many of the same reasons: pesticides, habitat loss, monoculture agriculture, climate disruption. Nobody campaigns for the preservation of hoverflies. No social movement rallies to protect cacao midges. And yet, across vast parts of the world, when these insects are lost, the ecological and agricultural consequences are comparable to losing bees. The economic value of fly pollination, globally, has never been comprehensively calculated. The best available estimates, published in specialized entomological journals, place it in the tens of billions of dollars per year. In tropical regions, fly pollination may account for a larger share of agricultural production than bee pollination does. This is not a marginal contribution. This is a foundational ecological service. And it is being performed by insects that most of humanity, if asked, would classify as pests. The fly does not care what we think of her. She pollinates the cacao flower because the flower produces nectar she needs. She pollinates the mango because she is hungry. She visits thousands of flowers in her short adult life, depositing pollen, fertilizing reproduction, maintaining the botanical foundation of ecosystems across every continent, without being asked, without recognition, for as long as flowering plants have existed. Chapter 4, The Decomposers, What Flies Do for the Planet Every Single Day. Imagine for a moment a world without flies. Not a reduced population, not scarcity, complete absence, no flies of any kind anywhere on Earth, from one day to the next. The cacao industry collapses within a season. Tropical fruit yields begin to decline within months. Certain flower species start to disappear. Maggot therapy ceases. Fundamental genetic research slows across dozens of fields. But these are the second-order consequences. The first-order consequence, the one that would affect every ecosystem on the planet, including the ones humans live in, is something else. Death would start accumulating. When an animal dies in the wild, any animal from a mouse to an elephant, a specific sequence of biological events follows. Bacteria begin multiplying within minutes. Rigor mortis sets in. The body begins to generate gases, swell, and change color. And within hours, usually within minutes, in warm conditions, flies arrive. Blowflies are often the first to detect a dead body. Their olfactory receptors can identify the chemical signatures of death from over a kilometer away. They arrive. They lay eggs. Within 24 hours, the eggs hatch. Within 48 hours, the larvae are actively consuming the body. A single fly can lay 300 eggs on a single carcass. Multiple females arriving in sequence can deposit thousands. The resulting larval biomass, processing dead tissue, accelerating decomposition, recycling organic material, is one of the most efficient bioprocessing systems in the natural world. Entomologists have quantified this. A small mammal carcass in warm weather is reduced to bone and minimal residue by fly larvae in approximately 7 to 10 days. Without flies, the same process would take months and would release the organic material back into the environment in forms that attract other scavengers, pollute water sources, and potentially transmit disease. Flies are, in this context, the most rapid and comprehensive organic recyclers on Earth. They process the dead animals, the excrement, the rotting fruit, the accumulated organic debris of every ecosystem they inhabit, and they do it at a pace no human technology can match. One thought experiment makes the scale clearer. Biologists have estimated that without fly-driven decomposition, if all flies disappeared and the decomposition work fell entirely to bacteria, fungi, and other scavengers, the accumulated organic matter produced globally in a single year would be sufficient to alter the chemistry of terrestrial ecosystems measurably. Within a decade, the cumulative impact would be transformative. Dead biomass would build up, water systems would be contaminated at a scale modern sanitation infrastructure could not absorb. Disease organisms, normally consumed by fly larvae and destroyed in their digestive systems, would persist in the environment. The planet would not become uninhabitable. Life would continue, but the systems we rely on, agriculture, water supplies, public health, would degrade in ways that would take decades to correct. This is not hyperbole. This is the documented ecological role of a group of insects most people regard as vermin. And the work is continuous. Every moment of every day, across every ecosystem on Earth, flies are processing organic matter. A dead bird by the side of a road, a deer hit by a car in the forest, an apple rotting in a field, animal waste on a pasture, all of it continuously returned to the soil by the digestive systems of fly larvae. Without this processing, the nitrogen cycle itself would function less efficiently. Soil fertility would decline, plant health would suffer, agricultural productivity would drop. The fly is not just cleaning up, she is participating at planetary scale in the ongoing maintenance of the nutrient cycles that all terrestrial life depends on. The vulture processes the large carcasses. The fly processes everything else. Together, they are among the most important sanitation systems ever produced by biological evolution, and both have been historically treated as threats rather than partners. The fly has been doing this for hundreds of millions of years, longer than flowering plants existed, longer than most mammals existed, longer than almost any other insect group on Earth. She predates the work. She invented the work, and we step on her without thinking about it. Chapter 5, Forensic Science, Space Exploration, and What Flies Taught Us About Life. In 1910, a German biologist named Thomas Hunt Morgan began working with a tiny species of fly that had infested fruit left out in his laboratory. The fly was Drosophila melanogaster, the common fruit fly. Morgan began breeding them, observing them, mapping the patterns of inheritance from generation to generation. Within 5 years, he had discovered that genes are located on chromosomes. Morgan received the Nobel Prize in 1933. His discovery began a scientific relationship that has continued for over a century and that has produced an extraordinary list of achievements. 6 Nobel Prizes in medicine or physiology have been awarded for work done primarily on Drosophila. The identification of genes responsible for body development, the discovery of how the immune system recognizes foreign pathogens, the mapping of genetic circuits that control biological clocks and circadian rhythms, research that has fundamental implications for our understanding of sleep, metabolism, and age-related disease. Every one of these breakthroughs originated in the study of a fly. The reason Drosophila became so central to modern biology is straightforward. Flies reproduce quickly. Their genetics are simple enough to study systematically. Their short life cycles allow researchers to observe multiple generations within a single experiment. And, crucially, they share approximately 60% of their genes with human beings. When a scientist studies a genetic disease in a fly, they are often studying, at the molecular level, the same mechanism that causes the disease in humans. Research on flies has contributed to our understanding of cancer, of neurodegenerative diseases like Parkinson's and Alzheimer's, of diabetes, of sleep disorders, of the biological basis of addiction, of how memories form in the brain, of why organisms age. Some of these insights would have been impossible without flies. Their reproductive speed makes possible experiments that would take centuries in larger animals. And yet, almost no one outside specialized scientific communities knows any of this. The Drosophila sits in laboratories around the world, in carefully controlled conditions, under microscopes, in experimental apparatus, performing some of the most important biological research in human history. Flies have also gone to space. Drosophila were among the first living organisms ever sent into orbit. They have been aboard the International Space Station for decades. Researchers have used them to study the effects of microgravity on cellular biology, immune function, heart development, and cognitive behavior. Nearly everything we understand about how biological systems respond to the space environment, data that is foundational to planning long-duration human spaceflight, has come partly from flies. Before humans can live on the Moon or Mars, flies will have lived there. They already have begun preparing the way. And there is one more domain where flies have transformed human knowledge. Forensic science, the timing of insect colonization on a dead body, which species arrive first, how quickly larvae develop, what temperatures and conditions affect a sequence, allows forensic entomologists to determine the time of death with remarkable precision, sometimes down to the hour. Evidence from fly colonization has been used to solve murders, to identify the locations where bodies were originally killed, to establish whether remains were moved after death, to exonerate the wrongfully accused, to provide closure to families who might otherwise never have known what happened to their loved ones. This work is conducted in collaboration with the flies themselves, studying their life cycles, reading their development stages, interpreting what their presence indicates about the history of a scene. The insect that most people kill on sight is, in this specific context, a witness, a recorder, a source of truth that humans could not access without her. Genetics, medicine, space biology, criminal justice, these are not marginal applications. These are among the most consequential fields of modern science, and the fly has contributed substantially to all of them. We have not built statues to her. We have not named institutions after her. We have not commissioned textbooks that reflect her importance. We have instead continued to treat her as a pest while benefiting every single day from knowledge she has been providing for over a century. The paradox? Disease vectors and the cost of misunderstanding. This episode cannot end without an honest accounting of what flies also do. Some species of fly transmit disease. The tsetse fly across parts of Africa transmits trypanosomes, the parasites that cause sleeping sickness in humans and agana in livestock. Sandflies transmit leishmaniasis, a disfiguring and sometimes fatal disease affecting millions of people in tropical regions. Black flies transmit river blindness, once one of the leading causes of blindness in rural Africa. Common houseflies can mechanically carry pathogens from contaminated surfaces to food, contributing to outbreaks of diarrheal disease. The harm is real, it is not trivial, and it falls disproportionately on the poorest populations on Earth. But honest accounting requires proportion. Of the 150,000 known fly species, the number that transmits significant human disease is in the dozens, fewer than 1 in 1,000. The overwhelming majority of flies pose no direct threat to human health at all. The cacao midge does not transmit disease. The butterfly does not transmit disease. The fungus gnats pollinating plants in cold climates do not transmit disease. The forensic blowfly that arrives at a body in a sealed room does not transmit disease. The Drosophila in a laboratory does not transmit disease. When we extend our fear of a small number of disease vector species to the entire insect order, we are making the same categorical error we made with rats, with vultures, with snakes. We are confusing a local problem with a global identity. The comparison with other disease vectors is also worth making. Mosquitoes kill approximately 700,000 people per year, primarily through malaria. Flies, across all disease-transmitting species combined, cause a small fraction of that mortality. We do not categorize mosquitoes the same way we categorize flies. The word mosquito carries a specific, narrower connotation, an insect associated with blood feeding, with itching, with summer nights. The word fly carries a diffuse, generalized negative charge that extends across the entire order of insects. This is a failure of vocabulary more than a failure of observation. We simply do not have the cultural categories to distinguish between disease-carrying flies and the vast majority of fly species that contribute to ecosystems, pollinate crops, and heal human wounds. None of this is an argument for tolerating flies on food. Hygiene matters. Sanitation matters. Protecting prepared food from contamination matters. The historical battle against flies in hospitals, in kitchens, in agricultural settings has genuinely reduced disease transmission and saved lives. But organized extermination of all flies, treating them as categorically harmful, ignores what the scientific record actually shows. Targeted management of disease vector species is different from blanket hostility toward an entire insect order. Public health measures that address specific transmission pathways are different from the reflexive killing of every fly that lands on a surface. Education is different from annihilation. We have built multi-billion-dollar industries around killing flies. Insecticide sprays, fly swatters, sticky traps, electric devices, chemical repellents, a global pest control industry worth tens of billions of dollars annually, much of it directed at species that perform essential ecological work. We have built, in other words, an economic system that depends on the continuous elimination of insects that we simultaneously depend on for pollination, decomposition, and medical research. The contradiction is rarely noticed. The fly has not asked for our permission. She has not waited for us to understand what she does. She has simply continued, pollinating, decomposing, healing wounds, contributing to the genetic research that has saved countless human lives while we swatted her by the billions. She has absorbed our ignorance without complaint because she has no mechanism for complaint. She has paid for our misunderstanding with her life again and again for as long as we have been killing her. And she has kept working. The insect you will never swat the same way again. The next time one lands on your arm, stop. Not to welcome her, not to keep her there, simply for 1 second before you react to see her clearly. That insect has compound eyes with 4,000 lenses, processing the world at 4 times your visual speed. She has gyroscopes in her body more sophisticated than the ones in modern aircraft. She can taste what she is standing on through her feet. If she is a hoverfly, she is one of the most important pollinators in your region. If she is a blowfly, her species is cleaning up organic matter in quantities that would otherwise poison water supplies. If she is a fruit fly, her genetic simplicity has made possible decades of medical research that underpins modern cancer treatment. If she is a green bottle fly, her larvae are being used right now in hospitals to heal wounds that nothing else can heal. She is not asking to be loved. She is asking, by her very presence, to be seen for what she actually is, not a symbol of dirt, not a carrier of disease, except in a small number of specific species. Not a nuisance, an insect that has been working silently in the background of life on Earth for hundreds of millions of years. We have spent centuries building fear around her. Entire industries profit from that fear. Entire cultures transmit it from one generation to the next. And beneath the fear, consistently, has been a scientific record that tells a different story. The fly pollinates the flower that becomes the chocolate a child holds in her hand. The fly decomposes the dead leaf on the forest floor, returning its minerals to the soil that grows the tree. The fly cleans the wound of a diabetic patient who would otherwise lose a leg. The fly dies in a laboratory after contributing data that will, in some future decade, help cure a disease that afflicts someone not yet born. She does not need our gratitude. She does not experience recognition. She has no concept of thanks and no use for it. Scopes in her body more sophisticated than the ones in modern aircraft. She can taste what she is standing on through her feet. But we can, at minimum, try to see her accurately, to understand what she is doing, to make peace with the fact that the insect most of us have spent our lives trying to eliminate is, by almost every measure that matters ecologically and medically, one of the most valuable organisms on Earth. The next episode of Wild Purpose, an animal that most people picture as a threat, a large mammal, powerful, feared across cultures for thousands of years. A creature whose silhouette alone is enough to change the atmosphere of any forest scene. An animal whose ecological role is so fundamental that the forests she inhabits literally would not look the same without her. Who spreads seeds across hundreds of square kilometers. Who carries ocean nutrients into inland ecosystems. Who, in her absence, causes entire food webs to reorganize. The bear. This was Wild Purpose.